Hello, everybody. I'm here today with our science project. My name is Donovan Ludovina, and our science project was testing the efficacy of active ingredients within skincare products. Today, I'm also joined by Adryanne Gonzalez and Cameron Strobis.
What's up?
Introduction. For our project, we'll be using a Kirby Bauer test on each skincare ingredient, along with the two actual products. The zones of inhibition from each ingredient will be determined to see if there's any effectiveness in eliminating the bacteria. And our hypothesis that we developed was when the skincare products ingredients were tested, the benzoyl peroxide would have the highest zones of inhibition.
So, the basis of our research was looking into propionic bacterium, which is one of the main causes of inflammation for acne. This is an opportunistic pathogen which can sometimes just be dormant and sometimes can cause diseases. It is 1 to 2 micrometers in size and is gram positive. It typically goes into sebaceous filaments or hair follicles onto the skin and grows in anaerobic environments. Next slide, please. The second part we looked into was the actual active ingredients themselves and what they claim to do. So some remove dead skin, dirt, and sebum. Rather than actually kill bacteria, they exfoliate the skin, preventing the environment from happening. And others actually aim to destroy the bacteria. Those that claim to kill the bacteria are the benzoyl peroxide, sulfur, and tea tree oil. The ingredients that clean the sebum, dirt, and dead skin are the salicylic acids, alpha hydroxylic acids, like lactic acid. Sorry.
So our materials that we used in this project were 1% benzoyl peroxide, 1% salicylic acid, tea tree oil, lactic acid, the Buffet by the Ordinary, 30 milliliters of LB broth, liquid culture of E. coli. The specific E. coli we used was HB101K12. We had a cell spreader, 360 milliliters of LB agar, an autoclave, sterile inoculating loops, 18 petri dishes, filter paper discs, ampicillin, and 70% isopropyl alcohol. For our methods, we used 100 milliliters of LB broth, which we prepared and sterilized in an autoclave, and then we used a sterile loop to put the E. coli from the liquid culture into the broth. The broth was then placed in the incubator to allow more of the E. coli to grow overnight, and then 360 milliliters of the LB agar will be prepared and sterilized. 18 petri dishes will be labeled and sectioned into quadrants for each of the products, and then once the agar was cooled, then 20 milliliters will be put into each petri dish. 1% solution will be made for the benzoyl peroxide and the salicylic acid, and the tea tree oil, the lactic acid, and the Buffet will be used as is. Enough filter paper discs will be made in order to have 10 trials. The filter paper disc will be soaked in their respective solutions for up to 5 minutes each. 1 milliliter of the previously made bacteria culture will be pipetted onto the agar plates, and utilizing a cell spreader will be dispersed evenly throughout the entire dish. The discs will also be placed into their respective quadrants, and the plates will be put in the incubator overnight to allow for that growth. The positive control of this experiment is the ampicillin, which will show the result when the product will have worked. The zone of inhibition, which is the zone with no bacterial growth, can then be measured, and the negative control will have no zone of inhibition and will have a full lawn of bacteria. Once the bacteria has grown, the plates can be examined, the zones of inhibition can be measured, pictures can be taken, and the plates will be sprayed with 70% isopropyl alcohol, then disposed of into the biohazard trash.
So our conclusions and our results. Up to this point, we were not able to get any really substantial amount of plate growth, and so we were unable to report any results. This happened because of the 10 trials that we had. There was not enough growth on the plates, so we are unable to report, like I said. But we will be working on this in the meantime to get actual substantial results for the project.
Thank you for your time.
Thank,
Thanks, folks.
thank you.